Another reminder on these attendance sheets, you have to put your first name and last name. You have to fill out your student ID number up here at the top, and then the bubbles that are associated.
Once you start talking, I'll sit for a
0
bit.
through 9. If you don't do that, the system will not register you actually being here, and that's no fun. Also, there are 5 questions at the bottom, 1 through 5, that you can answer while the talk is going on. But to answer those, you can't just simply circle them down here. I need you to fill up the corresponding bubbles up top. So questions 1 through 5, fill the bubbles up here, because again, the software is not going to track your responses if you don't do that. But that combination of attendance and your answers helps us just keep track of everything. So appreciate you doing that. And at the end of seminar, you'll just bring these sheets back down to me before you go. Sound good? Alrighty. So let's go ahead and get this party started. Welcome to Science Seminar. Are we excited?
Yeah.
Woo.
That's not acceptable. We can do better than that. Are we excited, friends? We're gonna. All right. This will take time, but I'm going to work you into this. I'm really excited to have Bree and Matt joining us today from Bionanomics. So they've come over from Electric Works. If you're not from Fort Wayne, you're not familiar, it's just down the road here. People call it the GE campus a lot. And they're with this Bionanomics group, and they're going to talk to us about that today. And so let's hear it for Bree and Matt as they join us. Thank you so much.
Yeah. Thanks for inviting us, Dr. Beachel. We're really excited to come talk to you guys today. So as he said, we are Bionanomics. We are a nonprofit biomakerspace downtown in Electric Works. We're a very new organization. There's only 4 of us, truly. And you're looking at, we've got 3 of us here. So today we're going to talk to you about cells, how we code them, and how we help the community by doing so. So I'm Bree. I'm the lab operations director. That's Matt. He's our lab automation director. And I'm going to take you through today's plan. So as long as we don't run out of time, these are all the things that we're going to talk about: introductions, overviews, research, programs, and then we'll do a closing. We do have a slide at the end of this talk. We're going to give you a link to this slide deck because all of the pictures and stuff that we've put in have links to them that you can click and find out more information. So I will give you a link to that. We have a link for if you're interested in our internship program, link to the last talk that we gave last year. So there's lots of good information. We'll make sure to get that to Dr. Beachel and out to the rest of you guys. All right, so here we go. Again, Bree. I am really interested in microbiology. I got really interested in it because as a kid I was watching a bunch of zombie movies, and I was always really excited like of those CDC field researchers that were like researching the cure against the clock, and I thought that was really cool. I was like, I'm gonna do that one day. Turns out you have to be a medical doctor to do that with extra schooling, and I got through college and I was like, I'm done. That's it, never again. So I didn't do that. I just did microbiology and immunology over at PFW. I graduated in 19. And then I went to work at Lutheran Hospital in their clinical lab. I did the microbiology section. We do a lot of like setups for cultures, for like wound cultures or urine cultures, blah, blah, blah. So I mess with a lot of bodily fluids, and I'm glad I'm out of there now. But I was there for about 5 years. And now I am the Queen Lord of the lab over at BNX. That was a title given to me by somebody else. I would never give that to myself. That's obnoxious. But I am also a mom. I have a 4-year-old girl. She's a cutie patootie. I'm really into arts and crafts, Legos, games. I try to mesh those two together. This auger art. I did that all by myself. I made that for like a Halloween contest, and so the black is Proteus vulgaris, and then the green is E. coli, and this is on like a Hectoan auger, which is just a pretty color. So I did that all by myself. Look at me go. And these are my dogs, Pancake and Cinnamon. They're so cute. And here's Matt.
Hey, so I'm Matt McDonough. I kind of got into the biology space first through getting into botany. I've been kind of just traipsing around the ecosystems here in Indiana for the better part of the last 5 years with me and a bunch of friends. Kind of got into macro photography and taking really close-up macros of flowers. One thing kind of led to another. I bought a scanning electron microscope off Craigslist and ended up moving it into our lab here, and kind of all just went from there. And now I do a lot of our like microscope maintenance, and I'm in charge of some of our automation goals surrounding the microscopy stuff and our experiments there. So that's kind of how I got into all this. I'm going to pass it back to Bree here for a bit to explain more of our company's stuff. So
Thanks. So, like I said, we're a nonprofit. We got started, we were incorporated as a business officially in July of 24. Sorry, I'm distracted. But I was hired on before that, trying to get things going. The founder, CEO, president, Doug Horner, is actually the CEO of a medical software company called MIE. He came into some money and he wanted to spend it doing biology stuff instead of buying random stuff. So that's what we did. And we started a lab, and it's kind of like his side quest. But for me, it's the main quest. Anyways, so what he likes to say is that we do robotics at every scale, microbes to machines. So if you look at some of the like machinery that we do for like the aircraft, this is an aircraft and this is a flagellar motor. And you can see the huge similarity between those two things. So he was really interested in how that worked. And so he brought me on to help figure it out. We have a pipeline that we have. We are two-armed, so we have the biology arm and a robotics arm. I'm not going to talk much about the robotics arm today because we're a biology seminar, not a robotics seminar. But anyway, briefly, we have our pipeline. We start with kids in high school that are going through our AMP Lab collaboration program doing robotics, and we also work with students that are in the robotics competition. You guys may have heard of it, FIRST, FRC, whatever. And so we teach them the engineering and coding skills that they need to learn going into college, and then we steal them as interns, and they help us build things to help automate our biology lab. Of course, we also take biology interns, so hit me up if you're interested. But our goal is to automate the entire biology lab 99%. So there's very little human interaction. And obviously that helps with repeatability, accuracy of our experiments. And it's just also super convenient because you can just press a button and walk away. So that's the goal. It's going to take a while to get there, but that's the goal. All right, so what do we research? The ultimate goal is to do some cell fate determination. So what decisions a cell makes when they divide into like a neuron or a muscle or whatever. So your stem cells, what drives that decision is what we're trying to figure out. So there are a few different things you guys may or may not have learned about. I don't know, is this like all grades? Okay, cool. So you may have learned this, you may have not. But so I'm just gonna briefly go over. We have a couple of things. So we have transcription factors, which are proteins that latch on upstream of the gene that either inhibit or promote the transcription, hence transcription factor. We also have physical properties of the genome. So when you have it methylated or wrapped around in a histone, the protein complexes that transcribe cannot get to this, but they can get to this. So this is another one of the ways that determines the cell decision. We also have cell-to-cell signaling, and there are a bunch of different kinds. This one is direct intracellular signaling, where they send a molecule like a buddy who's already differentiated, sends them the signal, and then they differentiate as well. And a subsection of that is morphogen cellular signaling. So basically this is concentration dependent. So you have the source of the signal here, and then depending on the concentration gradient is what makes the decision to turn it into basically the neurons, the muscles, and like the epidermis. So there's like a cutoff there, and then they differentiate like so. So we wanna, those are some of the things that, oh, I forgot, my bad. This is based on Michael Levin's research. So this is a bioelectric signal. You guys may have seen planaria. They are the model organism that a lot of people research for like regeneration studies. Basically, if you cut this guy in half, the head side will grow a tail and the tail side will grow a head. And that's like their fission reproduction and not the sexual reproduction. So this experiment it cut it into thirds, and then it submersed this middle section into like a really high strength ionic chemical and made it grow two heads instead. And then they let that grow. They cut it twice in just water, and each time, instead of growing a tail, all of them grew two heads. So something here, the ionic signaling made like neuronal memory happen here. So that is a really interesting piece that Michael Levin was researching, and that's kind of inspired Doug to do some of the things that he does. So what are we gonna do to figure out cell fate determination? So we're gonna try to sequence it, which is a pretty advanced technique. It's a little complicated, but I'm gonna try to break it down for you. You guys have seen DNA to RNA to proteins. It's everywhere all the time. So what you can do is sequence the DNA, the genome. You can sequence the RNA, all your transcripts that come from the genome, and then you can sequence the proteins that come from the RNA. Not all RNA gets translated into proteins. You've got your rRNA, tRNA, blah, 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 blah. And in fact, most of them don't turn into proteins. It's important to differentiate those two. In my head, it was really hard for me to differentiate. So proteins have different lifetimes than RNA. They go through their post-translational modifications. The translation efficiency varies, so you can't just do a one-to-one, oh, RNA straight to protein. So that's why we sequence those differently. Our lab is mostly going to focus on DNA and RNA. So, basically what we'll do is we'll take cells at each division and sequence their genome and their transcriptome and see what is changing as it divides, what genes are getting transcribed, what is being methylated, et cetera, and just figure out what's driving that change. And then once we figure it out, we can try to reverse it, replicate it, manipulate it, blah, blah, blah. Why do we care? We care because it is super impactful for a bunch of different areas of biology. So cancer research, it can help with the progression of disease. It can help you learn about the metastasis, blah, blah, blah. And it can help with, I'm blanking. Why am I blanking? Awkward. Let me go back. Oh yeah, birth defects, stem cell therapies for diseases, and also evolutionary divergences among species. So you can use the DNA to tell when this species differentiated from that species. Thanks. So we also have interns. We are quite a new organization, so we don't have many interns. But these are some of the works that they've done so far. So we do biology stuff and computer science stuff. So Ashwin Riker is a PFW grad. He's in his master's program, and he is doing an AR. Is it going?
Here.
Do I just need to go
It's
to
a
the
second
browser?
monitor, so you gotta.
So he's doing an AR VR simulation where you can grab a protein and move it around and look around at its like 3D structure. I like to throw them. I don't know, it's fun. So we have a bunch of different molecules on that so far, and he's got size information and function information there. That's what he's up to. That app is on the Meta Store for free if you guys wanna, if you happen to have a Meta Quest and you wanna download it. This is Jaden. He was an intern last summer, and he was doing the techniques for time-lapsing embryos, which was kind of hard to figure out, to be honest. So this is a time lapse of an embryo as it grows of C. elegans, which is a nematode. Super, super widely used organism. And you can see as it turns from cells to its worm stage, and then it hatches. There it goes. He has moved on. We miss him very much. We also have some Carroll High School.
Oh shoot, sorry.
That's okay.
Did this one first, I guess.
That's fine. We have Mike Delaley, who's actually an Indiana Tech student, and he is doing biofilm research with Dr. Beachel. This is a picture that he took very recently. He posts most of his pictures and stuff on his Instagram, which is linked here, so check that out. But this is of Strep mutans biofilm. The red are the bacterial cells. The blue are the sugars they create. No, I'm so sorry. The blue is the sugars. I have that backwards. I'm so sorry. The green is the cells. The red is the EPS, and the blue is the sugars. Should have looked at my notes. And then we're back. Sorry, Mike. We also have some high school students from Carroll that are doing colorectal cancer research on Drosophila. So I have a picture of that where they were dissecting it. Not my cup of tea, but that's what they like to do. Moving on. We also do a program called iGEM. It's an international synthetic bio competition. I'm not going to talk about it very much because we're doing a call-out meeting next week, which I also have linked at the end of the talk, but I'll briefly go over it. Basically, you get a group of students together and you try to pick a local or global issue and solve it with synthetic bio. It's a multifaceted project, so you have outreach, engineering, the wet lab stuff, so you need a multidisciplinary team. This is an example of one of those projects, how you have the wet lab components on, ooh, my back. I'm so sorry. And now it's not there. Sorry, Matt.
All good.
Spoiler. You have the wet lab components on the left side, and then you have the outreach at the bottom right, and then you have the engineering component on the top, which is like they developed the container that the assay would go in for field work. They 3D printed it, CAD'd it, etc. And then the wet lab people designed an assay to detect algae in their local rivers. So that was a Poland team, and that's an example of what we do. Again, we're having a call-out meeting next week on Wednesday in Dr. Beachel's lab that you can learn more. So with that, I will turn it over to Matt.
How's it going? All right, let me get around here. Okay, so I'm going to talk a little bit about our program called Open IDI. This is a program basically dedicated to building communities to answer questions that we still have about the natural world, e.g. the real world that surrounds us. So I'm going to depart from this program for a minute and just rant about native ecology here. We're going to loop it back, don't worry. But I want everybody to start kind of thinking about ecology as a living machine, because it more or less is. It's a machine that in most of the US has taken about 400 million years to get to the state it has gotten to today. And every little piece, every microbe, every animal is a cog in that machine. And that's what I want us to start kind of thinking about this analogy. So what's important when we're maintaining a machine? We need documentation on all the parts of the machine. We need experienced engineers that know what they're doing. We also need to pay attention to the machine. Is it making like a horrible squelching sound? Is it operating cleanly? Are the gears greased up? So yeah, things we need to pay attention to about our ecology, just like we would about a machine. So how is our machine running in Fort Wayne, Indiana right now? So a lot of it's gone, just to put that out there. But when you drive maybe from home to work, you're going to be seeing a lot of this kind of stuff. A lot of just strip malls, parking lots. Not very much, actual nature anywhere in there. And that's kind of the experience of living in the greater Midwest as a whole. You're just kind of driving between maybe a turf grass lawn and a parking lot every day. And how are we actually exposed to the plants that are in our parking lots? A lot of them are just random ass plants from halfway across the world that are grouped together based on aesthetics, have basically no ecological or pollinator benefit to any of our native insects and stuff like that. We also tend to plant a lot of really invasive trees. So Bradford pears, this is a screenshot from one of our local CVS's here. Just Bradford pears, which are a horribly invasive tree lining the side of the road. It's a pretty common sight here. And then a lot of us, when we're done existing in the public spaces, we drive back home and we see a turf grass lawn at the end of the day. And I don't know, maybe, some people like turf grass. That's, if you like it, if you want a little patch to play soccer, I think that's fine. But when I see this, I kind of think of just a flat Minecraft world. It kind of doesn't make me feel good at all. It doesn't, just doesn't get me very excited. It doesn't, and it provides no benefit to any of the native ecology. So if we look at two of these yards here, the yard on the left, I don't know. If I look at that, I kind of feel like I'm about to get yelled at, kicked out of the neighborhood. It just doesn't imbue a sense of calm to me. Maybe like it does for some other people. But the yard on the right, I can hear all the bees buzzing around. I can hear the insects. You could stand in that yard for an hour and probably see 30 different species of insect just hanging out on those flowers. I think that's pretty cool. So why are we putting turf grass in our yards, and what even is it? So in a lot of the U.S., we're putting Kentucky bluegrass down in our yards. And Kentucky bluegrass is an interesting grass, which we'll get to in a second. But one thing about it is that its root systems remain really dense and really compact. So this image on the right here, we can see that's about what a Kentucky bluegrass lawn would look like. And you've probably experienced this if you ever move turf around. If you just roll it up, it's going to roll up like carpet. I mean, it comes out fairly easy for a plant like that, where all of our native shrubs, they're going to have root systems that extend 2 to 3 times deeper than what you're seeing above. So all of our like prairie plants, we have a lot of herbaceous perennials in Indiana, and a lot of our native plants, you're basically just seeing the tip of the iceberg when you see the plant. It's shooting a lot of roots down every year when it grows, and the root systems stay alive every year and the top halves die back, which also makes these really effective carbon sinks. I don't know if you guys are familiar with the concept of carbon sinks here, but a carbon sink is basically any natural or man-made system that pulls carbon out of the air and stores it, which is good, because I don't know if you've heard, but we have a little too much carbon in the atmosphere right now. But native lawns are actually really good at being carbon sinks. And on top of that, when you have turf grass, it's basically like concrete. Like water will hit that and just roll right off. It's going to cause a lot of flooding compared to a soil that's much more highly aerated. So this is my little lawn rant, but we're going to loop it back, I promise. So why do we use Kentucky bluegrass? I mean, it's named Kentucky bluegrass. They named the music after it. It's got to be an American grass, right? That's actually a European and Asian grass. So when the settlers first brought it over, it grew really well in Kentucky's limestone-rich soils, and that's why it's called Kentucky bluegrass. It's actually not native to this region of the world at all. It's naturalized here at this point, but it's not native. So, yeah, why are we doing this? Why do we keep planting our yards with what is essentially ecologically just patches of concrete. I think it's honestly just because it's what we're used to. We don't really know any better. So I'm trying to start getting you guys thinking about what is better, what's a better way to do it. So which of these two pieces is better for maintaining that machine we're talking about? The picture on the left here, we basically ripped all the cogs out and thrown them away. And the picture on the right, we have restored some cogs that were originally there and put them back. And so our machine's going to be functioning a little bit better if we have more patches of land like this. And just as a reminder, this machine is the machine that makes our air breathable and stuff like that. So it's an important one to maintain. But what came before all this? Like what was Indiana like 200 years ago, so to say? So this is a picture actually from 1938 to 2026. It's a time lapse of the North Coliseum area. So as we end of the modern times, you can see some of the stores you're probably familiar with shopping at. But even 100 years ago, we had some patches of forest maybe that were very fragmented by cornfields. You can see a lot of fields in these early images. So even 100 years ago, our machine was sort of fragmented. It was sort of broken up. And if we look here, kind of freeze frame here, we can kind of see a good example of that. So just a lot of little fragmented ecosystems here. And it's worth noting that even the ecosystems you're seeing in this image, I mean, humans had already been settling this area for 150 years before. And that's not even modern, European humans, I guess is what I'm saying. So all these forests would have been flat plowed or they would have been logged and plowed before, even before these images. So all the forests in this picture are probably only, 50 to 100 years old. We actually only have 2 old growth forests north of Indiana, or north of Indianapolis in Indiana still. We'll get to that in a second. But one thing I want us to start thinking about is just connecting to Indiana as a place instead of just like where our houses and where our jobs are. So one thing I hear a lot, and one thing I'm guilty of thinking quite a lot, especially as a kid, is that Indiana is just cornfields. It's really boring. I mean, and it honestly is pretty boring to drive through if you pick the wrong road. So you're probably just going to be seeing corn all day. But Indiana historically was not ever this boring. So Indiana used to be about 88% forest cover and a lot of wetlands and interspersed dry prairies. We had some oak mesic savannas up here. And in Fort Wayne specifically, if we see this little green cut or blue cutout of wetland there, this was actually the historical remnant of the Great Black Swamp, which was a swamp that ran from where we're standing here today all the way to Lake Erie. It was one contiguous ecosystem that would have been pretty much the Everglades of the North. So there was a lot of really cool species here, and there still are if you sort of look in the right cracks. So, sorry, I don't know how, I think the mic was too far from my mouth, sorry about that. So what's actually in Indiana? We have a lot of really cool stuff in Indiana, from really rare spiders to really rare plants. And one of my favorite casts of plants in Indiana are our carnivorous plants. So we actually have pitcher plants, these guys up here, these eat bugs. This is a native Indiana species. You can find these in Fort Wayne, actually, if you know where to look. Same with Drosera rotundifolia right here. It's a sundew, but it works kind of like a Venus flytrap. If a fly lands on that, it kind of closes up around it and eats the fly. And those are just a few. We have so many really cool species in Indiana that are still here if you know where to look. So what does Indiana's machine look like right now? It's a bunch of fragmented pieces of what once was a fully contiguous system, basically, right? But a lot of these fragmented pieces are still in pretty good shape, and we like to refer to these as remnants. So what is a remnant? A remnant is a piece of land that has largely evaded human development for the better course of modern human history. So we're actually fortunate enough to have one of these up in Huntertown, just about 20 minutes north of here. This is Bicentennial Woods. You might have been there before. It's a pretty popular hiking trail in the area. But this forest, more or less, has never been logged. So everywhere else around it has been logged, but there's trees in here that sprouted during the Civil War and are still growing today. You can go touch them. Some huge, for the biggest sycamores you'll ever see in your life out there. And this is one of 2 old growth forests that is remaining north of Indianapolis. The other one's in St. Joe County, up near South Bend. But we're pretty lucky to have a remnant like this so close to us. I encourage you guys to go check it out. But when we think about like what's going on with the land around it, this is the only spot of land that hasn't been developed on in the last, 300 years. So there's stuff here that has been extirpated from everywhere else in the state. So when we start looking at these remnants, we can find species here that we might not have even thought existed in Indiana, but once we start looking, we say, oh, like, well, how the hell did this get here? It must have been here before. And we'll get more into that in a second. But remnants are important because they represent pieces of this machine that are still functioning as they're supposed to. So we have disjunct groups of cogs that are still spinning the way they're supposed to. They might not all be connected like they used to be, but more or less, these are still functional pieces of our ecosystems. And so where do we find remnants most often in Indiana? We actually, if we look at this map, this is that North Coliseum map again, but I want you to kind of pay attention to where things aren't changing. And that's mostly along our rivers here. So rivers, kind of hard to build a house on top of. So we don't tend to do that a lot. And if we look at all of the rivers and all the wetland areas, the wetlands that we haven't developed over tend to host some species and some casts of some really rare plants. And so I'm going to talk about one of those plants for just a minute here. So this is a flower. It's called Phacelia covellii. It's a scorpion weed, and it's one of the only scorpion weeds that grows out in the Midwest. And this is a plant that we're trying to figure out how it actually got here. So if I look at this graph here, this range down here is the traditional range of this plant. A few years ago, my buddy Evan Bennett was out hiking. He's a great amateur botanist here in Indiana, and he was kayaking along a watershed up in this region, or along the St. Joe River. Pulled off his kayak to have some lunch, and saw this flower growing in the brush. So he took a picture of it, posted it to iNaturalist, and didn't really think anything of it. You know, a few months later, we had all these people commenting like, "What the hell is this? Like, this isn't supposed to be here. This is an Appalachian flower." And since we started looking for it up here, we found it in 5 or 10 different locations up here. So we're finding this flower that traditionally only grows in the Appalachians that has jumped a couple hundred miles all the way up here. And we just kind of want to know, like, how did it actually get up here? Like, how did a plant that has, you know, seeds that are about as tiny as a sesame seed and aren't wind dispersed and aren't animal dispersed make the jump all the way across all this landmass? So that's one of the questions we're trying to answer with this plant in particular. And we thought, you know, maybe we had a bird that got it stuck on its leg. We've been thinking about theories of how this could have gotten up here. So we thought maybe there was a professor that studies this plant that suggested to us that there might be like really microscopic glochids or hairs on the base of the seed that could attach to like a bird leg. We were actually able to run the seed through our scanning electron microscope and found that that was not the case. So answered one question, but we have a lot more. So basically, looping this all back to our program OpenIDI, our program OpenIDI is built to answer questions like this, like how the hell did this plant move 600 miles? How does it exist here now? It's a program to answer these questions that we have about the ecology around us in a deeper way than just like throwing it on iNaturalist. Basically going to go into OpenIDI really quick. If you've used iNaturalist, I based the UI a little bit off iNaturalist for our Explore feature here. This is a screenshot of our software that runs this program though. So we can see the flower there and where it was collected. If I click into the software, we can see all the collection information, where it's at. It's currently, you know, in our lab. Here's the locality notes and the habitat. And then you can also access all of the imaging and you know, DNA work we've done on the plant in raw format. So we actually built a SEM raw image viewer into the website where you can go in, you can zoom into like, you know, the micrometer and nanometer scale and take measurements and export images. So we're basically taking the tools, like we'll do the collection work, we'll handle your images, but once we get the images done, it's back in the hands of the citizen researchers that are doing this to try to make their own discoveries with. So the Phacelia project is one we're working on, the one I've been kind of going over here through this program. The other one we're working on a couple right now. We also found this species called Virginia mallow. That's this plant right here. This is actually another Appalachian species. The closest place it really grows is in the Ohio River Valley, and we keep finding populations up around Ann Arbor, Michigan. So another one of these plants where we're trying to figure out how the hell did it move so far? Was it always here? You know, just trying to answer these questions. And then the image on the right here is from a study we're doing with a guy named Wolfgang Blum, and he's a super like high caliber cactus researcher out in Germany, I think. But he we're taking a bunch of high quality scanning electron microscope images of a lot of the spines in this cactus genus. This is an Echinocerius spine, because he thinks that there might be a way to differentiate some of these species that are almost impossible to tell apart without DNA work on the microscopic level based on the morphology of the spines. So that's another fun project we're working on taxonomically. We're also jumping a little bit beyond taxonomic research, and we're poised to help answer kind of some of the deeper questions that you might have during the taxonomic research. So Bree was already talking about these flatworms earlier, the ones you could cut in half and they'd grow 2 heads. And there's a link here when you download the slide deck if you want to read more about that. But there's another thing in nature that kind of already hijacks these cellular signaling pathways that we're pretty interested in, and that is plant galls. So if you've never seen a gall, well, you probably have seen a gall, maybe you just didn't know what it was, but they're these structures that grow on plants that just kind of look out of place. And these form when an insect, usually a mite or a wasp, lands on the plant. It will inject a peptide or an enzyme into the plant, and then that actually is able to kick the cells back into a stem cell state, hijack their genetic pathway, and then basically grow a house for its larva to develop in. And they come in, millions of different shapes and forms, and there are tons of species that have evolved this ability. But plants kind of already can hack, or bugs can already kind of hack plants in the way we want to hack cells. So we're interested in studying them because we think that there might be some interesting cellular signaling stuff going on at that level as well. So we're partnered up with a guy named Adam Kranz out in New Mexico. He's probably one of the nation's foremost gall researchers. But we're going to be running some trials with him where we induce some peptides that he's designed onto the root tips of Arabidopsis thaliana, which is this plant up here. This is from our phase 1 trial where we're just trying to keep these alive for a long time under time lapse. We're still kind of working out our methodologies surrounding that. But this image here is just like a 6-hour time lapse of the plant dying, which still looks kind of cool. But once we get this worked out, our next goal is to induce some peptides that he's designed into these cells and time lapse how they respond. We're going to be doing a lot of experiments around that. And then there's another layer after that where once we see how those are responding, we're going to be trying to measure electrochemical gradients across the material as these get induced to see if we can listen in on the crosstalk that the cells are doing when that gall induction starts. So that's kind of a brief overview on OpenIDI and one of our big programs we're launching into at our lab. I encourage all of you to kind of get involved just in your local ecology, and the best way to do that is just by starting to notice things around you, while you're walking to class or walking to work or, anywhere you're out and about. Just try to learn about this giant living machine that surrounds you, and try to see, does this piece belong here? Is this from another country? How does this work? And it's pretty enriching once you get into it. We have a link to OpenIDI here, and also iNaturalist. iNaturalist is great if you just want to start getting to know what's around you. There's an explore feature. You can drag a square around your house. You can say, what insects have been seen around my house? Really fun to get into. I'm sure some of you in this room have probably used it. And yeah, so if you want to get more involved with any of our stuff, there's those links, and then we got business cards on the table, QR codes to this presentation, and also some other fun stuff in there. So I think that's about all we got for you in terms of presentation today, but what?
Q&A.
Yeah, questions if anybody has them.
Any questions from anybody? So I wanted to just wanted to wait to the end of your presentation just to kind of open the floor up to you all, because you have an opportunity here to step into a lab and do research with, really passionate people that have a lot of incredibly nice equipment. And that availability is not common. And so usually you have to be in graduate school or some type of professional master's program or whatever to have access to this. And so to be an undergrad student and to be able to pick up a project, you can see no matter what you're interested in, they got a little bit of everything going on, whether it's, human side, animal side, plant side, macro scale, micro scale, genetics. They have everything there for you. And so the door has been opened, and I would encourage you to walk through it.